Hi guys. So today I would like to just give an introduction lecture. This is largely going to be geared for the Masters 651 Biochemistry students to kind of, for maybe if you're a little bit rusty on your carbohydrate chemistry, just to give you a nice introduction to where we're going to lead. Or for my Biochemistry 2, undergraduate Biochemistry 2 students who perhaps, again, maybe did carbohydrate biochemistry a little while ago, or perhaps their professor didn't focus too much on carbohydrates, and so you'd like to get up to speed. And in that respect, it's geared towards getting students to understand the chemistry of carbohydrates in order to better understand for the 651 students, to better understand both glycobiology, which I consider as being kind of where carbohydrates influence biology, and particularly in terms of how carbohydrates may modify molecules to make glycoconjugates of proteins or lipids, and also how carbohydrates, of course, feed into glycolysis. Or similarly for my Biochemistry 2 students, where the carbohydrates are going to, again, feed into glycolysis. But this is really just some of the basic chemistry of carbohydrates, just to prepare that target audience in particular. So what I would like you guys to understand is actually really just these top three, or actually no, rather just these top two. And these bottom three are going to be what I would follow up with in the class with our formal lectures. But really I'd like to introduce to you guys the structures and the names of monosaccharides. So mono, one, saccharide, sugar, so just the single sugars. And also understand the open chain and ring forms of those monosaccharides. And this then leads into what happens when those monosaccharides link together to form disaccharides, so two of them linked together, or the polysaccharides, where there may be several of them linked together to make a chain, kind of like the chains of peptides made up of amino acids. Sugars, the same kind of thing. These little bricks can come together to form larger structures. And then, like I say, with 651, and also with my 451, but to a lesser degree, representing the graduate and undergraduate levels respectively, we're going to talk about how these molecules are important in modifying things like amino acids and lipids in order to modify biological functions, but that'll be for the lecture material. So to start out, let's just define carbohydrates. Now they're named because unlike lipids, which are going to be lots of CH2, CH2, CH3, the end of the chain, CH3, so kind of alkyl chains, and these alkyl chains can go on and on and on and can be very long, like we discussed with the fatty acids, and these are going to be very hydrophobic for the lipids. Rather, in contrast to that, the carbohydrates, they're still based upon this carbon, but they have water molecules, H2O, across the carbon. So rather than it being CH2, it might be CH, OH, and then we have the CH2O. And because of that, it's going to be a lot more hydrophilic rather than rather hydrophobic. And that's going to have a large impact. Also, it's going to change the reactivity of this carbon because it's bonded to an oxygen. So in general, these carbons are a lot more reactive. However, they're also more oxidized, which is important when it comes to taking energy from these carbons in order to use them for respiration reactions, and underlines really the reason why why lipids have more energy per mole, per gram, than carbohydrates, to do with the oxidation state of this carbon here. But I digress. Really, it's just that the basic understanding is that it's the C H2O rather than just the CH2 of the alkyl. They may have come from a variety of different places, but on the planet Earth, at the moment, the majority of carbohydrates come from the photosynthesis of carbon fixation from CO2 and water in the Calvin-Benson cycle element of photosynthesis. And like I said, they start out as monosaccharides, so just one sugar, and you may be very familiar with the structure of glucose like this here, but they can link together to form chains, polymers, disaccharides, polysaccharides. And they can start out rather small, like for instance glyceraldehyde or DHAP, which you would see in glycolysis, in step 4 glycolysis. But they can chain together to form much larger and even absolutely huge polysaccharides, such as starch, and of which amylopectin is a constituent, or glycogen. To go further, the biological function of these is, of course, as an energy source, and of course energy storage. So energy source of glucose that's going to enter into glycolysis, or energy storage of the glycogen and starch, but also quite often structural. So cell walls of certain organisms will use glucose in order to rigidify their cell wall, such as in plants, the cell wall, cellulose. The cell wall has got cellulose that gives it that rigidity. Or chitin, which you find in many organisms, especially like small bugs, so exoskeletons. Or in the bacterial cell wall, actually, there are carbohydrates that help to rigidify the structure. So there's also structural and also information molecules. So, for instance, in cell-to-cell signaling, something I talk about in some of my classes and some of my lectures, is the idea that these sugars get added onto lipids of the lipid membrane, or onto carbohydrates, or onto proteins. They're embedded into the lipid membrane, or even within the cell itself, and can help and be responsible for cell-cell signaling. For instance, blood typing, blood type A, blood type B, blood type O. Those are all given by the chains of sugars added to ceramides on the cell membrane of red blood cells, erythrocytes. And so the differentiation between blood type A, blood type B, blood type O is actually just the arrangement of sugars which you have, whether or not it's going to be a certain chain of one type or another, or neither for blood type A, B, O. So it's actually important for cell-cell recognition. What tells the difference between a pathogen versus a healthy cell? Your immune system can recognize by the chains of sugars which are attached to the outsides of your cells. It kind of acts as like a key. So there's a variety of different ways. And of course, keeping in with that, yes, sugars can be linked to proteins to form glycoproteins and proteoglycans, which are subtly different that I'll talk about in the lecture, but not to do with this introduction here. Now, of course, one of the main things we think of with sugar is that sugar is sweet. And so why is sugar sweet? And we have these receptors, there's TAS1R, 2, and 3 receptors, which are G-coupled protein receptors, which are embedded integral proteins attached to plasma membranes. They are receptors. And what will happen is that a sugar can come in, can bind to this receptor, cause a protein conformational change, which causes a downstream effect of activating the GTP binding protein that causes a further signal transduction within the cell that is then converted into a neuronal response, and our brains interpret that as being sweet. So that's where that comes from. And so something needs to be a ligand for this protein in order to stimulate that response. And there are various sugars that do that, but also the sweeteners can also do that and can also bind to this receptor and activate it and create a stimulating feeling of, this is sweet. But the way in which it binds, as with all molecules, the ligand needs to bind, and so it needs to be a good fit for the protein. And part of that fit is also going to be the 3D orientation in space. It needs to be a steric match. It needs to actually match. And a lot of time we get used to just looking at the 2D structure of things, just flat on a piece of paper, because that's just how we read them and experience this in the classroom. But in reality, none of these things are 2D, they are all 3D. And so although something may have the same kind of chemical empirical formula, and maybe even you may have the arrangement on 2D may appear very similar, in 3D they're going to be very different. And one of the key things we'll be looking at actually is chirality. And you can have between the L and the D version of aspartame, is going to depend on, is going to influence whether or not that molecule is going to bind to the binding pocket of the protein, and whether or not that ligand is going to bind correctly to stimulate the downstream response. And it's going to be the same with all of the sugars. You have all of the different, you have the stereoisomers, enantiomers and things like that, and that's going to influence the way that it binds. And this is going to be really, really important to biology. Although it may not look like much, if you have a sugar where maybe the OH is on one side versus the OH being on the other side, although to us that may seem like nothing, it actually makes a huge difference. And so for that reason, we're going to look at quite a lot of stereoisomers when it comes to sugars. But first, a little bit of background in order to bring that into place. So the sugars, you're going to be used to seeing them in the cyclic form, which was from that first slide, which is going to be, oh, I've kind of butchered this a little bit. It's going to look something like this, and you'll be used to something that looks like this, CH2OH, and it's going to have this projection like so to illustrate the 3D shape, and it's going to look something like this. And that's the cyclic form, which most people are really familiar with. And just a note as well, some of these OHs, I guess I should draw in to make it clear, draw the hydrogens to be clear. Some of these OHs are pointed upwards and some of them pointed downwards. And that's not by a coincidence. That's actually very important. And exactly the same as on the previous slide, we had left versus right, and they were, although they may have the same empirical formula, and although you may think, hey, what's the difference? It's just putting the OH to the left or to the right. Surely that doesn't make a difference. Actually, it does. It makes a big difference. It's going to influence a lot about the chemistry and the downstream application in the biology. And in the same way, up versus down is going to influence the way that the sugar kind of interacts with the biology. And exactly the same as we had with the receptor, down versus up is going to influence how it might bind to a receptor, and you may not think it's a lot, actually it ends up being quite a lot. But what we learn is that with the sugars, you guys will be used to them in the cyclic form, but they also exist in the linear form, and like I had a few slides ago, and what happens is that the linear form cyclizes to the cyclic form. And it's something that's in equilibrium. Being an aqueous solution, favors it being in the cyclic form. Being dry favors it being in the linear form. But when we look at the rules of stereochemistry, we look at it in the linear version, first of all, because it's clearer to see what's going on. And it's clearer to then apply that to the cyclic version. And so we're going to start out looking at the linear versions of the sugars before finishing up talking about how we cyclize them, chemical reaction of the cyclizing, and then how that may have changed things a little bit. So first of all, I'm going to introduce the two main groups of sugar. It's actually the aldoses and the ketoses. And the aldose family of sugars are called aldoses because the functional carbonyl group is an aldehyde, where you have an aldehyde is characterized by a carbonyl, which is a C double bond O, that is bound to a carbon chain, an R. It could be of any kind of length, or it could be H actually. Although that's just for aldehydes in general. It couldn't be just an H for a sugar, because then it would just be acetone. But it's going to be bound to a carbonyl. It's going to be bound to an R group, aliphatic. But then it's going to have the other bond is going to be with a hydrogen. Whereas in contrast with the ketones, you see the carbonyl is bound to two R groups. So an aldehyde is this, and a ketone is R1, R2. So that's the difference between an aldehyde and a ketone. And the sugars can be divided into aldoses and ketoses, depending upon the linear structure of them. And for instance, this has got 1, 2, 3 carbons, so it's an aldotriose. This has also got 3 carbons, so it is a ketotriose, tries for 3. And for instance, when we go up to a lot of sugars are hexoses, so it's got 6 carbons. A lot of sugars in biology and biochemistry have got 6 carbons, so it would be aldohexose or ketohexose, but these examples here are trioses. And notice as well that the OHs are to the right this time, but there's nothing stopping them being to the left, or one to the right, one to the left, top one to the left, bottom one to the right, top one to the right, bottom one to the left. They're going to be completely different sugars. This one here, DHA, and if it's got a phospho group on it, it would be DHAP, DHAP, which is the DHAP is the one of the sugars that is produced in step 4 of glycolysis that you might be familiar with, but that's for another day. So for the aldoses, there's a whole bunch of them, and you can see that they all have this aldehyde. Carbon number 1 is an aldehyde. 1, 2, 3, 4. 4 carbon sugar aldose here. Carbon number 1 is an aldehyde. There's a whole bunch of them. There's 6 carbons. 1, 2, 3, 4, 5, 6. And carbon number 1 is, again, an aldehyde. On the other hand, the ketose is carbon number 1, 2, 3. And carbon number 2 is the carbonyl, and this is a ketone group instead, where you have the R group is attached to both sides of the carbonyl carbon. And fructose being probably the most famous ketose there is. There's fewer of them in biology. They come up less often in biology, but nonetheless are still very, very important. Now we're going to move over and talk about the stereochemistry here. So stereoisomers, we can broadly kind of differentiate them into a few different categories of the stereoisomers. You have the enantiomers. These are those mirror images, those ones that revolve polar light. Those are the ones with the handedness, with the left hand versus the right hand. And you can kind of think of it as, well, like I say, handedness. And if you think of maybe some like action movie or something like that, and there's the main character has to scan their hand to get into a secret room or something like that. And if you get your hands and put them down in front of you on your desk in front of you, and you look, and on your right hand, your thumb is to the left, and on your left hand, your thumb is to the right. And so if you were, perhaps there was some sort of hand scanner, and you had to put it down, and it recognized your right hand with your thumb to your left, that's no problem. But if you were to get your left hand and put it down, then your thumb's to the right. And so it's not going to fit onto the hand scanner correctly. Let's just pretend they've got some primitive hand scanner that can't compensate for that. Whereas if it's expecting to have the thumb on the left side, then the thumb needs to be on the left side. If you, from your right hand, if you put on your left hand, then your thumb's to the right side. It's in the wrong place, so it won't recognize it. Receptors are going to be in the same way, and although my hands are maybe very similar, they are mirror images of each other, and I can't superimpose them. I can't put them on top of each other in a way where my thumb is in the same place. If I was just rotate my hand and make it so my thumb is in the same place, well, now it's the front of my hand versus the back of my hand, and that's not the same thing. On the other hand, with the diastereomers, these are different arrangements of atoms in 3D space that are not mirror images of each other. You have you can divide those up into the geometric isomers, which are going to be conformers, which are just different arrangements around single bonds. Similar with the atropic isomers, which are going to be arrangements around a single bond, but ones which are sterically hindered, and so it kind of is as if it was a double bond, that kind of thing. Or the easy, which is that cis and trans, which is to do with where you cannot rotate around the double bond. And this type is not really relevant for sugars. But what is relevant for sugars are these epimers and anomers. And epimers is just that idea where if you have the different, say a carbon chain, and you have the OH on one side versus being on the other side, that is going to, those are going to be different epimers of each other. And in some cases, there may also be an element of chirality there. mirror images that are associated with the enantiomers. And when it comes to the enantiomers of sugars, we actually just really discuss the penultimate carbon as being I mean, there may be several enantiomers, but it's only really the penultimate carbon that we worry about with sugars in a biochemistry, biology point of view. From a strictly chemistry point of view, yes, there may be other enantiomers, chiral centers, but we only worry about the penultimate carbon as the chiral carbon. Epimers are just going to be where you swap the OH to the right or to the left or up or down, and that's going to create different molecules where it may have the same empirical formula, but it's actually going to be a different molecule because of the arrangement in 3D space. And anomers, which is a special type of epimer, which is to do with one carbon in particular, and actually it's going to be what we call the anomeric carbon. It's going to come in a little bit later with the cyclization. And again, whether or not that OH is going to be pointing up or down, that we'll get onto in a little bit later. But first, let's talk about enantiomers. So enantiomers are stereoisomers that are non-superimposable mirror images. Like I said, in sugar, there are several chiral centers. So you know, the handedness is going to be over a chiral center, and a chiral center is a carbon which has 1, 2, 3, 4 different groups attached to it. So if I had CH2 and then let's say R1 and R2, well, you know, although it's got 4 different things attached to it, hydrogen and hydrogen are the same thing. So it's only got 3 different things. There's 4 groups in total, but only 3 of them are different because hydrogen and hydrogen are the same. So that would not be chiral. But if it was, for instance, carbon with R1, R2, R3, and a hydrogen or a R4, then this has got 1, 2, 3, 4 different groups. So it'll be chiral, and we have to give it a little star to say that it's chiral. And those are the ones which are going to produce the non-superimposable mirror images of each other. And when it comes to biochemistry, we only really think about, in the carbon chain, we only think of the penultimate carbon, that's the one before last, as being the one that's got this chiral center which is relevant for in the biochemistry. And it's designated as being in the D or the L form, whether it's the OH is to the left, making it D, or whether or not it is to the right, making it L. And this D and L may be unfamiliar, but it's actually the same as S and R. It's just that it comes from a more ancient place, not a more ancient place, but an older place. And it's just that when biochemists first started talking about sugars, they would compare the sugars to glyceraldehyde, and rather than calling it the kind of clockwise and anti-clockwise with the plus and minus and the R and S, they had it with the dextro or levo, which is Latin for right and left, respectively, and they just kind of named it like that, and it's just kind of stuck. So it is actually S and R at the same time as being D and L, but we just actually we just tend to use D and L instead. That's what people do. If you start talking about S and R, people are probably going to look at you funny in the terms of biology and biochemistry, but it's not strictly wrong. So these D and L isomers are going to be are enantiomers of each other. They're talking about whether or not it's going to be clockwise or anti-clockwise in terms of priority that comes in later, and to do with whether or not the OH is to the left or to the right. And most hexoses, so that's 6 carbon sugars in life, they actually usually have the D stereoisomer. So unlike in amino acids where it's usually the L, and we also use the L and the D rather than the R and the S in amino acids, rather than it being the in the amino acids it's L, in the sugars it tends to be the D. Now, a lot of people will say it's all. It's actually not all. Some prokaryotes do have the L sugar instead, but 99.9 times out of 100, it's going to be the D isomer, and that's the one that I'm always going to talk about. D-glucose is the one that humans are going to be using in glycolysis, And in most cases, we're talking about the D isomer, such as D-glucose. And so, like I said, we're talking about these mirror images, where if we were to get the, this molecule here, this sugar here, this is actually glyceraldehyde, and we were to contrast it and look at the mirror of it, you have one orientation versus the other orientation, and they are non-superimposable mirror images of each other. And if we look at it in a 2D format, it's going to look like this with glyceraldehyde. And in our naming formats, we name oxygen as being priority number 1, hydrogen is priority number 4, and we're going to go around priority number 2, we're going to go around this way for the D sugar, or if the OH is on the left, then we're going to be, this is priority 1, this is priority 4, this is priority 2, so we're going to be going around this way. So the D is where the OH is on the right, for dextro, right, and L, the OH is on the left for levo, left, and it's going to rotate around this way. So this is the D sugar, and this is the L sugar. But the D sugar is the one which is most common in biology. And similarly, if we just look at the hexose glucose, 1, 2, 3, 4, 5, 6, we can see that we have the mirror image of the sugar being on the right versus the sugar being on the left. And although it may not look like much, and you may think, hey, this isn't such a big deal, it actually is a really, really big deal and completely influences the way in which a sugar can act as a ligand for a receptor or another protein, and it's going to completely change the interaction. And you can see that in the D glucose, you have on the right side, you have these 3 OHs, and then the carbon number 3 is on the has the OH on the left, whereas with the L, you've got the 3 OHs and the carbon number 3 is on the right. And when it cyclizes, that's going to change the way that it's orientated, and it's going to change the way in which it may fit into a receptor. And so if, for instance, you have a ligand sugar, which has got group A, group B, and group C, and it's got to bind into the protein in pocket A, pocket B, where group A binds to pocket A, group B binds to pocket B, group C binds to pocket C. That's going to align really, really nicely if it's the correct enantiomer. But if it's the not the correct enantiomer, and we were to swap the A and the C, it's still got the same empirical formula, still might appear very similar, and whether or not the OH maybe is on the left versus the OH being on the right. And you see now that group A now aligns with pocket C, so that doesn't interact properly. And group C lines up with pocket A, and so that doesn't line up properly. So it's not going to, they're not lining up properly, so it's not going to bind. You may think, well, hey, let's just rotate it around. But if you rotate it around, it doesn't make it any better. You can never rotate it in order to make it to fit. Exactly the same as with my 2 hands. There's no way I can rotate this hand to make it so it superimposes over the other hand with my thumbs in the right place. Because even if I do it where I turn it like this, I've now got the front of my hand versus the back of my hand, or the palm of my hand versus the back of my hand. And so it's not going to be the same. And think of it again as that example I gave with like the hand scanner. Let's say the hand scanner is only compatible with your thumb being on the left of your right hand. And you put it down, it's going to read your palm, read your fingerprint, and read your thumbprint, and it needs to have the thumb on the left. But if you think, well, hey, with my left hand, I'm just going to be smart and just turn it around so that my thumb is on the left. Well, yeah, your thumb's on the left, but it can't read your fingerprint because it's the back of your hand. It's going to be exactly the same with the sugar. And so that's why the enantiomers and just really the stereochemistry in general is really important for the sugars. Now, on the other hand, we have the diastereomers. Diastereomers are non-mirror images of sugars, and it's where you're going to have a different arrangement of the OH groups that doesn't create a mirror image. And whereas with the kind of chiral mirror images, the physical properties, so the melting point and solubility, doesn't change. On the other hand, with the diastereomers, the physical properties such as solubility and melting point, they do change. For instance, between etherose and therose, the OHs, here we have this carbon number 2, so it's not the penultimate carbon, so it's not that mirror image one, where that makes it a D or an L sugar. With carbon number 2, if I move it from the right to the left, that's going to actually create a different sugar. It's still both D sugars, but we consider it a different sugar. These sugars are diastereomers of each other, and they're going to have different water solubilities, for instance. So between, for instance, glucose, mannose, and galactose, these are all hexoses with 6 carbons, but you see that between carbon 2 of mannose and glucose, you can see that you've got the right-handed OH versus the left-handed OH, glucose versus mannose, and they're going to have different physical properties. Likewise, with the glucose versus the galactose, carbon number 4, you've got the OH on the right versus the OH on the left. Now, these diastereomers, where the only difference between glucose and mannose, and the only difference between glucose and galactose is just the position of the OH on, or the configuration around one carbon atom, those are called epimers. Epimers are just where you change the configuration around one carbon, just for one of the carbons, you switch the OH from the left to the right or the right to the left. Those are epimers. So glucose and mannose are epimers of each other, and glucose and galactose are epimers of each other, but not galactose and mannose, because there's 2 things that are different between these ones, and so that would be 2 carbon difference, so they wouldn't be epimers of each other. But it'd still be diastereomers with different physical properties, but not epimers. And epimers is going to lead in a little bit into the anomers later on when we talk about cyclization. We don't see it with the straight chain form, and you'll see why in a minute. So to recap, enantiomers, these are the stereoisomers that are non-superimposable mirror images. Diastereomers are non-mirror image differential arrangements of groups. So enantiomer is going to be the mirror image. It's going to be the D versus L around the carbon before last. Whereas with the diastereomers, this is going to be a non-mirror image, not around that chiral center at the penultimate carbon. And these are going to have the different physical properties, whereas the epimers are going to be differences around one carbon in particular, just one carbon, which is going to be switching the OH from the left to the right or the right to the left. So the key points to take from this are 2 families of monosaccharides. Those are the aldoses and the ketoses, and that depends on whether or not the carbonyl is going to be carbon number 1, carbon number 2, or whether or not, if it's carbon number 1, it's going to be an aldehyde where it's got a hydrogen attached to it, or it's an aldehyde, or whether or not it's going to be a ketone where it may have the R group, but instead of having a hydrogen up here, it's going to be another R group, and this is going to be the ketos. So you have the aldose and the ketos, and monosaccharides are made up of unbranched carbon chains, single bonds linking all the carbons. One of the carbons is double bonded to an oxygen to form the carbonyl, and the other carbon atoms are bonded to hydroxyl groups. That's kind of the basic chemistry that we see. And like I said, if we take another look at the aldoses, there's the aldehyde group up here. You can see that yes, indeed, they are all aldehydes, and the ketoses, they are all indeed ketones. Carbon number 1, 2, with the aldehyde with 1, 2 carbons bonded to it. And that's all the way through these. I personally think that when it comes to learning these structures, memorizing these structures seems like a very trivial task, and so I don't really ask people to memorize these structures, although you will be required to, if I give you the straight chain, to cyclize it for me. But I think that if you were going to learn some sugars, perhaps to help you in your future careers, The main ones really that pop up in biology and biochemistry are going to be things like ribose, glucose, galactose, mannose, and fructose. These ones are the ones that come up the most. And you can see that glucose, galactose, and mannose, those are the ones that are epimers. And here we have mannose, which is, these are the ones that epimers. Fructose, of course, comes up an awful lot, and ribose as well for various different cellular processes that come up. This is an aldose. Fructose is probably the most famous ketose. So these are going to come up a bunch in biochemistry, and if you did want to learn some structures, these would be the ones to learn. Now, just to give you guys a little preview of what is to come in the future, is that I touch on this a little bit in Biochemistry 1, and I touch on it a lot in general biochemistry at the graduate level. I don't touch it on at all actually for Biochemistry 2. The idea that we have these kind of derivatives of sugars, such as if we start out here with glucose, you can change some of these functional groups, so change it from an OH to an amine to get glucosamine. So instead of glucose, glucosamine. You may have heard of glucosamine. It's like a supplement that maybe people take for their joints and things like that. And in Biochemistry 1, and especially in general biochemistry graduate level, we talk about why that is an awful lot. And these kind of heterosugars, where the OH has been swapped out for something else, such as an N-acetyl group, or a phospho group, of course, for the glucose 6-phosphate. That's now where it becomes more relevant for Biochemistry 2. We swap out this OH for a phospho group to make glucose 6-phosphate, or perhaps 1,6-biphosphate, or glucose 1-phosphate. Or you can get the carboxylic acid, you can get the lactone, which comes up in various processes. You can swap out the OH group for a methyl group, or even just a hydrogen on its own, which isn't shown here. And it doesn't have to be glucose. All of the sugars really can do this as well. And it comes up an awful lot. This one down here, sialic acid, comes up an awful lot, especially to do with hydration and extracellular fluid and things like that. And it acts as to attract water. Hyaluronic acid is one that you may be familiar with as a moisturizer, because it attracts a lot of water, which is something, again, I touch on kind of at the tail end of Biochemistry 1, just as a kind of a mention, but I do talk about quite a bit in graduate biochemistry. So just to kind of be aware of these, we're not going to go into these right now, but just be aware of these coming up. Now, like I said, the linear sugars, we've looked at the linear sugars, and we looked at the different forms of the linear sugars, but these linear sugars can cyclize to give you the cyclic sugars. In aqueous solution, a lot of the sugars, so aldotetroses, so that's going to be 4 carbon, but not the 4 carbon ketones, or the all sugars, which have got at least 5 carbons in their backbone. They're going to cyclize in aqueous solution, because this exists in equilibrium, actually. But the equilibrium in aqueous solution is heavily pushed towards the cyclic form. Although, remember, it's in equilibrium, so there's still going to be a small amount of the linear form, but it's heavily pushed towards the cyclic form. In the when these sugars are dry, though, it's going to be more towards the linear form. But we look at the linear form because it's easier to look at the stereochemistry and understand the differences between the sugars and the stereochemistry of the sugars in that way. And the chemical mechanism for that is important. And what we know is that these carbonyls here, oxygen is electronegative. It's hungry for electrons. And so a covalent bond is a sharing of a pair of electrons, or a double bond, 2 pairs of electrons, but sharing is not always equal. And so because oxygen is electronegative and it's pulling on those electrons, in the bond it has a greater share of the electrons, and it's going to have this slight delta, this D for Greek letter lowercase delta. It's going to have a slight delta negative, meaning it's going to have a slight negative charge because it's got a greater share of the electrons. And if that's going to be delta negative, well, that must mean that this carbon must be delta positive, have a slight positive charge. Now, the lone pair of electrons from the penultimate carbon, the penultimate hydroxy group, here, this can act as a nucleophile and attack this carbon right here, as we see here. It can attack it and then cause the hemiacetal that we see here, where this OH here is this oxygen here, and this carbonyl here, the oxygen, is this oxygen here. We're going to go into more detail about this in a little moment. I'm just introducing the general mechanism just to let you know why does this happen. Well, it's because the carbonyl carbon is delta positive and gets attacked by the penultimate hydroxy group, the lone pair of electrons, and the penultimate hydroxy group, which is acting as a nucleophile to attack it, and it's going to cause the a cyclization. You have the hemiacetal here, and I'm going to show you what hemiacetal is in 2 or 3 slides. Essentially, it's where the carbon has got 2 oxygens attached to it. I'm going to get onto that in just a moment, but gets this cyclic structure. Now, when they cyclize, it's going to produce 2 stereoisomers, where the OH can be above, or the OH below rather, or the OH can be above. Now, this again is going to be an epimer, right? This type of stereoisomer is an epimer, because it's just again similar to whether or not the OH is left or right, or up or down. That's going to be, and if we have one sugar where when it's cyclized it went down, and the other sugar is cyclized and the OH went up, well, those 2 are going to be epimers of each other, and in particular that's where that anomer comes in. These are anomers. Anomers are the isotopic forms of the monosaccharides, where the difference between them is around the carbon number 1, whether or not the OH is in the 3D space is that up or down configuration, and that makes it either the alpha or the beta form. If the OH is pointing down, it's the beta form. If the OH is pointing up, it's the alpha form. And so you have alpha glucose, and this molecule here is glucose. What they've actually got, it would be alpha glucose if the OH was, I apologize, I've got it the wrong way around. It would be alpha glucose is actually here, where the OH is pointing down, and beta glucose is where it's pointing up. I apologize, the beta is where the OH is up, the alpha is where the OH is down. Alpha is down, beta is up. So what they have drawn here is, this is glucose, and because the OH is down and the anomeric carbon number 1, this is beta glucose. And if the OH was pointing up, then it would be alpha. Alpha glucose. So beta, carbon 1 up, alpha, carbon 1 down. So this is the alpha glucose. This is alpha glucose. Now, like I said, these are hemiacetals. And you can have the hemiketals if there was the cyclization of the ketose sugars. And this is, like I said, it's where you get the reaction between an alcohol and an aldehyde in general. This obviously applies in this setting to the sugars, but overall, a hemiacetal and a hemiketal is where you have an alcohol reacting with an aldehyde or a ketone. And so if we go back here, what we have is, and where's my pen? Here it is, an alcohol reacting with an aldehyde. And so it's going to form a ketal. And a ketal is going to have the formula of the carbon. It's going to have OH, OR, H, and R. This is going to be a hemialdol, and I'll acetal. And this is, you can see that the carbon has got two oxygens attached to it, and then it's got an R group and a hydrogen. And if we swap that hydrogen instead actually to another R group, so we've got R1 and R2, then we hemi ketal instead. And what this means is that this carbon here, although it's not a carbon, it's not going to be delta positive because it's bound to this. It's got a double bond with an oxygen, which makes this carbon a delta positive and this oxygen delta negative. In the same way, this carbon has got two single bonds with oxygens, so that overall it has a delta positive charge. So it can be attacked by a second nucleophile, such as a second OH group from a second sugar. And in that case, you're going to form the acetal or the ketal rather than the hemi. And that's going to be the case when we come to link these sugars together as the O glycosidic bond for the disaccharide polysaccharide. That's something that's going to come later. But just to make sure that you guys are aware that the hemiacetal and the hemiketals, where you have the reaction of an aldehyde with an alcohol or a ketone with an alcohol, where this carbon here with its oxygen and its hydrogen, and it's of course it started bonded to one R group, but then an alcohol comes along and reacts with this carbon because it is delta positive, and the alcohol oxygen alone pair is acting as a nucleophile to attack it, and you get this linkage where you have an ether and an alcohol, and that is forming the that means that this carbon is not out of the woods yet. It's still bonded to two oxygens, which are both electronegative, both pulling on the electrons in the bonds, making the carbon still slightly delta positive, so it can be attacked by a second nucleophile. That's something I'm going to come on to in a little bit later. But that's the case for the hemiacetal, or for the same kind of thing with the hemiketal. It's just that instead of the carbonyl that you start with, instead of it being an aldehyde, it's a ketone, and so you have this R2 group. And you can see that across here to the right, where if I just clear the screen, you can see that there's our carbon here, bound to the R group as the carbon, and of course bounds to its carbonyl oxygen, which becomes this OH group down here. And in the cyclic form, there's the R group. And it has this, the alcohol comes along, which is the penultimate carbon of the, in the straight chain, the penultimate carbon where the hydroxy reacts, acts as a nucleophile and attacks. That's shown there in purple. You can see it comes in and it attacks and causes this cyclization, where now it is up in here. And like I say, it's going to produce two different versions, where the OH is down, which is the alpha, or the OH is up, which is the beta glucose, which we see here. And you can actually, biology and biochemistry, these can be changed and rotated, and you can have, there are certain reactions that can occur to swap it from one to the other, depending on the needs of the cell, which is an enzymatic process, which will be often covered later on in the course. But there is a way to swap around them, and it's called, the reaction is called a mutarotation. Now, when these cyclize, I've given the example of glucose and the aldose. It also is going to occur with fructose as a ketose, and it's going to do the same thing to produce the hemiketal, where you can see now the carbon has got is just bound to different things, but where are we up to? It's producing just a different molecule, where now instead of it being an OH, because the carbon has got CH2OH, instead of it being instead of going to form a CH2OH, it's going to be numbered as 1, 2, 3, 4, 5, 6, which is going to be 1, 2, 3, 4, 5, 6. And same thing, 1, 2, 1, 2, 3, 4, 5, 6. And with the same way, you can have the alpha and the beta form. It's the other way around in this case, but we don't really talk about fructose too much in my course. I mainly kind of stick to glucose. But just to make you guys aware, same thing happens where the penultimate hydroxyl can attack that delta positive carbonyl carbon and cause the cyclization. And I may notice as well, you see that it creates this 5-membered ring. Although it's still a hexose sugar with 6 carbons, it produces a 5-membered ring rather than a 6-membered ring. And we call those the pyranoses and the furanoses, where the 6-membered ring is a pyranose and the 5-membered ring is a furanose. We call them that because they structurally resemble pyran, and pyran is this molecule here. It's this 6-membered ring, hetero ring, which has got this sugar in here, or furan, which is this 5-membered ring that's got the oxygen here, and pyran and glucose and furan and fructose in the cyclic forms, they resemble them. They're not literally them, but they resemble them. And so we call the O's, means sugar. So if it's a sugar that resembles pyran, we call it pyranose. If it's a sugar that resembles furan, we call it the furanose. And the way that we name these is that if it's glucose, then it's gluco, and then when it's cyclized to resemble pyran, it'll be glucopyranose. So we split the name up. Fructose is fructose, but when it's cyclized, we refer to that cyclic form as being fructofuranose. So fructo means it's based upon the fructose 6 carbon ketos. Furan means it's going to, when it cyclizes, it resembles furan, and O's is just to say that it's a sugar. And it can, of course, like I say, be the alpha or the beta form, where with the pyranoses, the OH is on the bottom for alpha, and in the furanoses, the CH2OH is on the top for the alpha. But I'm not really going to push hard on furanose mutarotation too much, and animas too much. So now to finish up, I'm just going to talk about how we actually work out how you go from the linear form to the cyclic form, because that's also that's going to be important. It's important for understanding enzyme mechanics, and also understanding the chemistry and how they bind to receptors. But also it's going to be test material in my exams. So if we start out, what we call this structure here, the cyclic form, we call that the Haworth perspective. Whereas the linear form is what we start out with as the Fisher projection. And if we were to think of the Haworth perspective, think of it as being like we have this hexagon, and let's say that it's flat towards you like that, and let's just turn it. So now it's kind of facing you. And if you have these up, down, up, down, up, down, that's going to look like this, where it's kind of A is going to be like A. It's going to be pointing up as I take it flat, and then twist it towards you so it looks like that. And that's what you're looking at with the Haworth projection. It's saying, hey, imagine this thicker side is the one that's twisted towards you, and thus in the orientation, the OHs, this OH is going to be above, and these two OHs are going to be below. And so if it was to drop down into a receptor, these two OHs would be the ones that drop down and touch into the receptor, whereas the OH above is going to be kind of above it. And there's some rules that we have for drawing these, and I've kind of broken it down that I hope will help. So first of all, carbon number 1, If we go down, 1, 2, 3, 4, 5, 6, carbon number 1 for glucose, I'm just going to go through with glucose. Carbon number 1 is going to be, we can't predict from the Fisher projection, biology may influence, biochemistry might influence whether or not it's going to be the alpha or the beta configuration, but from a pure Haworth perspective, from a pure Fisher perspective rather, projection, we can't actually tell what the Haworth perspective is going to look like. So in an exam, you would be told to draw the alpha or the beta version because, in an exam, you don't know which one's which. In biology and biochemistry, there may be a preference for one or the other given by the biological conditions and the enzymes that may be present, and proteins that may be present in the chemical environment. But just going straight from the Fisher projection, we don't know. So when it cyclizes, the OH, carbon number 1, it's going to be up or down, and it's going to for the alpha or beta configurations respectively, and you'll be told which one to draw. The OHs in the middle, they're going to be either up or down, depending on whether or not they're right or left, respectively. You see that in blue, the OHs that are to the right are going to be down, and the OHs that are to the left are going to be up. And I kind of think of it as being that it just lies down like that. And so if I have this OH that is to the right, I guess it's right now this is my right, it may be coming up as a mirror image for you guys. But if I was to have this straight chain, I've got the OH to the right. If I twist it, now the OH, twist it clockwise 90 degrees, the OH is pointing down. Whereas if I have the OH on the left side, if I twist it 90 degrees, the OH is up. So the trick is that if the OH is to the right, it's going to be down. If the OH is to the left, it's going to be up, which you can see with the color coding here. And then finally, or not finally rather, but next, you have for converting Fisher to Haworth, if you have carbon number 5, the penultimate carbon, that's going to be right here, and the OH is going to be the ether carbon right here. And finally, for carbon number 6, you have the OH above, so you have the CH2OH, and that is going to, that's where, remember, this is where you have the, this last one, whether or not it's going to be up or down, is actually to do with whether or not it's the D or the L sugar. Remember that we talked about earlier. And if this one is in the, it's to do with this chiral carbon right here. And if this one is arranged so that the CH2OH is above, then that is the D sugar. But actually, it sometimes gets drawn where instead the CH2OH is below and here, and then this is an H above instead. And that's actually going to be the L sugar. That is not, you don't need to know that for undergraduate level, but you do need to know that for graduate level. That the D-glucose is going to be where the in the Haworth perspective, the CH2OH is above for D, and it is below for L. And that's just FYI for undergraduates, but test material for graduates. And so I've got step-by-steps. I'm not going to go, well, I guess, why not? I'll go through them right now, just real quick, but they're written right here. Step 1, just draw the 6-membered ring with the oxygen in the upper right. Step 2, number the carbons starting in the clockwise direction, starting with the anomeric carbon. 1, 2, 3, 4, 5, and CH2OH 6. I guess there should be a step 1b, which is to put in the CH2OH of carbon 6. Let's tidy this up a little bit. Let's still put it here, but I'll just be a little neater. Okay, so draw a 6-membered ring, CH2OH, Next, want to number them. 1, 2, 3, 4, 5, 6. Next, place the hydroxyl groups. So for the middle hydroxyl groups, so whether or not it's going to be down, up, down, where it's going to be right, left, right, as pointing down, left. Okay, yeah, right are going to be down, left are going to be pointing up. I thought there was something not quite right there. And then, okay, I apologize. Step 4 is going to be to place the CH2OH, projects upwards for the D enantiomer, downwards for the L enantiomer. So, OH, 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 up for the D, down for the L. And then finally place and that's something which you only have to do with the graduate level. With the undergraduate level, it's always going to be CH2OH in the D isomer. And then finally place the anomeric OH, which is going to be up for beta, down for alpha. And then that's it, that's the structure. So I would say, here is the structure of glucose, here is the structure of galactose, here is the structure of mannose. Draw the glucopyranose of this, or the alpha glucopyranose or the beta glucopyranose. That's going to tell you whether or not this anomeric carbon should be up or down. And I would recommend practicing this a lot because I like to use it in my exams and make it a high points question. And that's actually all I'm going to cover today. In class, we're going to go over glycosidic bonds, which is where these we go from the hemiacetal into the acetal by taking a second nucleophilic attack on the anomeric carbon here. That's going to be in the next lecture. But I just wanted to give you guys a basic kind of understanding of the chemistry first of all, which I hope will really, really help you. So thank you for your attention, and I'll see you soon.